Good evening. Good evening, ladies, gentlemen, distinguished guests from Kingdom, organizations, universities, community. Welcome to the keynote lecture by Professor Juan Izpisua Belmonte from Salk Institute. I'm sure, because I heard comments from people before this lecture, also in the previous days, there is a lot of expectations from this lecture. Being the theme time, many people interested, you can then reset time and, live forever, or, rejuvenate and whatever. Okay, this of course was a provocative way of announcing the scientific content of Professor Belmonte's contribution to WEP. And in a minute it will be clear why. Starting, in fact, from what is the scientific track, career, cultural track that Professor Belmonte has followed along the years, starting from the very early days of developmental biology, early studies in Italy for PhD in the University of Bologna, but then starting a long track of training in top labs that where people were discussing how embryonic development actually occurs at the molecular level. The early days when people started to dream of seeing how individual genes can contribute to complicated processes like morphogenesis, tissue regeneration, or reorganization of entire parts of the body of embryos in various model organisms. I think that's where Juan Carlos has contributed the most in the beginning to the foundation of now contemporary thinking. Now there is the era of stem cells, but then to understand the problem in the multiple dimensions. Stem cells regenerate tissues and so on, but all these complicated processes happen to occur in multiple dimensions. And time is one of the most important parameters, and we will learn this from his talk today, that regulate these complicated processes. So after the long postdoc in Heidelberg University and at the MBL, and then postdoc in University of California, Los Angeles with Eddie De Robertis. Then he started his own career at the Salk Institute, and Salk Institute, where he basically contributed to be one of the best top thinkers in one of what can consider one of the top places in the world for that matter. So we are particularly really excited, privileged, really excited, scientifically speaking, to have Juan Carlos here today. I'm sure everybody's going to really enjoy what we can call cutting-edge science in this such a hot topic. So thank you very much, Juan Carlos, for being here.
Good afternoon. Thank you very much, Valerio. It's a pleasure and an honor to be part of this program, and I would like to congratulate Valerio. I realize, and you all realize, that it's not an easy task to put together all the topics that we have been hearing and will be hearing in the next few days, and I am really honored to be here. Thank you, Valerio, for inviting me. I'm sorry for my voice. I got a small allergy. I hope I am understandable. Aging, getting old is not a good thing. It's a good thing on many aspects, but on the health aspect, it's not the best thing that it can happen to us. It's about 45 to 50 when all these major diseases, heart, cancer, diabetes, kidney disease, start to really hit the human being. You can see that image there around 50, that all the diseases start to go up. So the major risk factor for any disease is to get old, is when our cells, tissues, and organs start to deteriorate and they don't work as when we were young. So the idea here is either try to restore the function of these cells and tissues, or try to understand the process of aging so that we can postpone the appearance of these diseases or slow down aging. So I'll first dedicate about 10 minutes or so to the idea of generating cells, tissues, organs that can replace those that at a particular stage in our life don't work normally. And then I will dedicate the rest of the talk to genetic and epigenetic strategies to fix that problem. The idea of, as we age, our cells don't work the same because of natural aging, because of an accident, because of a disease. And this can be replaced by cells that we can generate in the lab. And that's what I call cellular strategies. It's about 25 years, more or less, that scientists around the world started to generate the different cell types that make up a human being in the laboratory. We are composed of about 250 cell types. And I will dare to say that in these 25 years of research, scientists around the world have been able to generate most of the cell types in the laboratory that a human being is formed of. Now, the question is why these cells are not used for transplantation if they are generated in the lab and they seem to be what they are, why are not in the clinic today? And perhaps the major question is this. Here you see a cardiomyocyte, a heart cell that is beating, that looks very similar to a cardiomyocyte that you will see in your heart and that has been produced in the lab. Why these cardiomyocytes, and we can generate millions and millions of them, why cannot be used today in the clinic for transplantation? And the problem is this. This is a cardiomyocyte that is produced by the human embryo in nature. And this here is the best cardiomyocyte that we can produce in the lab. They look the same, but they are not the same. Especially the late stages of maturation, they are difficult to mimic in the lab. And therefore, for a cardiomyocyte, for a hepatocyte, for a neuron, these late stages of maturation are not there yet. And that's the main reason why these cells cannot be used for transplantation. I'll mention briefly, as I said, for 5-7 minutes, an approach we are doing in the lab to try to solve this problem of maturation. And this has to do, and I apologize for some of you that were this morning in another talk where I mentioned this topic, but it will be very short. This has to do with the idea of chimeras. Chimeras is very common here in this culture. And you can see, for instance, in Egypt, here, one of the most famous things here is the mixing of two animals to give rise to a chimeric structure. So the experiment in the lab goes like this, is mixing cells from two different animals to obtain organs growing inside one animal. Here we have a blastocyst. A blastocyst is an early stage embryo, the earliest stages of embryogenesis. After the fertilization, the oocyte and the sperm get together, you get this blastocyst, and in red you see cells from another animal. The blastocyst is a mouse, the red is cells from the rat. And what we do is just inject these cells from the rat inside the mouse and look a few days later. And as you can see here, there is a mouse that has red cells all over its body. So the mouse provides a niche to have the rat cells be part of all the organs of this mouse. When the mouse is born, we don't know if it is a mouse or it is a rat. I normally make the joke that it's a mouse because we pay less per night of hotel for the mouse than for the rat. But really, we don't know what it is. It's a chimera of a mouse and a rat. Now, if we do this experiment many times, each time it's random. One day, in one experiment, the rat cells will be making part of the eye, will be making a heart, will be making a pancreas, but this is random. Now, if we are interested in obtaining a specific organ, what do? We do a small trick, And the trick is just to eliminate genetically the organ we want to obtain. Let's assume we want to obtain a pancreas that could be used for transplantation. What we do is we eliminate the formation of the pancreas in the host animal. We can do this genetically. We can eliminate, say, the PDX gene. When you eliminate this gene from the mouse, a pancreas is not formed. You get a mouse with no pancreas. If at the same time you introduce the rat cells, those rat cells will now colonize the empty niche, and they will make a pancreas inside the mouse. But in this case, will be a rat pancreas that now could be extracted from this mouse, that is just a reservoir, and could be transplanted to the rat that has a problem in the pancreas. This can be extended to almost any other organ. In here, you can see in this small embryo, this is the control on the left. Next to it, you see an embryo where we have genetically eliminated the heart, and that's why it's so small and it's not going to develop. But if now we introduce rat cells in red, you see that the heart comes back, and then the embryo develops normally. And that heart now could be transplanted back into a rat if that was needed. Another example, and I'll finish with that, with the eye. We eliminate the eye from the mouse, genetically introduce the rat cells, and now we have a mouse that has eyes from the rat that could be used for transplantation. So that's the concept that we're trying to develop in the lab. Using a host animal to introduce human cells to generate human organs for transplantation to replace those that stop working. There is one problem, and the problem is the following: the size of these organs. Here to the right, you have a wild-type rat. To the left, you have a wild-type mouse. In the middle, you have chimeras. But you can see that the size of the animal, the size of the chimera, is always the size of the host embryo. So if we introduce rat cells into a mouse, we are not going to get an organ the size of the rat. We'll get an organ the size of the host of the mouse, and vice versa. So if we were to introduce human cells into these animals, we will get probably organs, but they will not be the right size for a human to be successful. So that's why we decide to move to have animals as reservoirs that have an organ size that is more or less similar to the human being, and these are the pigs and the cows. In here you see a typical experiment where we inject 3 or 4 human cells in green into these pig blastocysts. You can see that these cells divide, and you have many more, and after a few days you have millions of human cells growing in the pig. You can see this better in this magnification. Now, the problem is that this experiment is not as efficient as the rat-mouse experiment I showed you before. We never get really an entire organ. So the way that the human cells talk to the pig cells is not as efficient as the rat talk to the mouse cells. There could be many reasons for this. The human and pig are evolutionary diverse. The evolutionary distance between a pig and a human cell is about 100 million years. Between a mouse and a rat is less, it's 20 million years. The time of gestation, to make an organ, to have a mouse be born, it takes 20 days, and a rat 21 days. So when we mix the cells, it's more or less the same time. Now, a human being takes 9 months, a pig takes 3 months. So to fit all these 9 months in 3 months of development in the pig, there could be certainly some constraints that will lower the efficiency. That's what we decide to use the host, the monkey animal. The monkey is an animal that is evolutionary closer to the pig, as you all know, and we're doing right now all these experiments. Now, I'm not going to show you the data, just suffice to say that the efficiency is bigger between human and monkeys than between human and pigs. Some of you will say, so what this person standing up there is trying to tell me that he's going to make a farm of monkeys to have each one of us a monkey for us to produce the organs that we will need later on in our life. I'm not saying this, and we are not doing this for that reason. First, economically, it will not be feasible. Remember that monkeys like us have only one baby compared to the pigs that have 15 piglets. So economically, it will not be really feasible. We may need more than one organ, so we will need many monkeys. But more important, ethically, it's something that probably we as humans, we will not accept in the near future. The reason we are doing these experiments is to try to learn the language that the human and the monkey, the molecular and genetic language, the human and the monkey talk, so that then we can translate this knowledge to the pig cells. So the situation is that we have a pig cell and a human cell that don't seem to talk to one another quite well. While the rat and the mouse, they talk quite well, and they can make organs between one another, the pig and human not. Now, we know that the human and monkey seems to talk quite well between one another, and thanks to the advances in the field of developmental biology and what are the key signaling pathways that make an embryo are now, we know a lot about that. We can modify these pathways that you see to the right of the skin in the pig cell according to what we learn from the monkey animal. And that's the reason we're doing this experiment in the monkey, to learn from the monkey and go back to the pig. And I hope in another occasion I can tell you a little bit more about this re-engineering of the pig embryo to be able to communicate better with the human cells so that we can produce human organs that can be used for replacement back into the human using the pig as a host animal. This is an approach that could help and benefit the dream of xenotransplantation. This is an area of research that has been with us for the last 40 years, and with the exception of a couple of papers that were published this year, not much advance has been made. And I feel together with the gene editing field that is developing at a really high speed, together these technologies of chimerism will help maybe to make this xenotransplantation a reality. But this is just expectations, and to be direct and with you, we still need a lot of knowledge to gather a lot of understanding before we can get this idea, even in the lab, let alone in the clinic. So I'm going to focus the major part of my talk in something that maybe is closer, this idea of using cells to somehow use them to replace those that stop working. It's, in my opinion, not in the near future. But what we think that is much closer is modifying our genome and modifying our epigenome to solve many of the problems that disease and aging causes in humans. And we can do these approaches, we can do it early on at the embryonic stage, or we can do it at the disease stage as an adult when the disease or the age is present. So let me give you a couple of examples at the early stage, at the embryonic stage. You know very well that the DNA is compartmentalized into structures, either inside the mitochondria or inside the nucleus. Mutations in the DNA, either mitochondrial DNA or nuclear DNA, produce and give rise to specific diseases. For instance, mitochondrial diseases. When a mutation is present in the mitochondrial DNA, this is transmitted to the next generation, and you will be amazed, for those of you that don't know, that is a high prevalence event. 1 every 200 mothers will transmit a mitochondrial mutation to their babies. The mitochondrial DNA is transmitted by the mother only. So this is a high number. And it happens that the mitochondria is the source of energy for our cells. So if there are mutations in the mitochondrial DNA, the level of energy of the cells will be low, the ATP levels will be low, and those tissues that really need a high level of energy will suffer. And that's why the kids that have this mitochondrial disease, most of them die at an early age because their muscle, their heart, their brain don't have energy and they die. So mitochondrial disease is a major problem for human beings, and the prevalence is quite high, as I mentioned in my numbers here. And one of the approaches we're following to try to restore the function of these mitochondria due to mutations in their DNA, it's based on this concept. So the disease only manifests when a big percentage of the mitochondria is affected. If you have less than 70% of mitochondria affected, the phenotype will not be that bad. But above a certain threshold, 60-70% of mitochondria affected in the cell, the phenotype will be obvious, and that will carry the disease. So the idea is not to fix all the mitochondria. If you just fix a few so that you lower that threshold, that will be enough because the phenotype will not be there. And how can we do this? And that can be done thanks to the new development in gene editing technologies. And I'm going to give you, as an example, this mouse. So this mouse is black and white in their skin. You can see this. And this comes from a cross of two mice that have a different mitochondria. And the only difference in this mitochondria is one base pair. The rest of the DNA is identical. So there is only one difference, one base pair different. And that's what makes this mouse black and white. So if we analyze the DNA of this mouse, we see two patterns of bands. We see one at the top. This is the DNA from one strain of the mouse, the black, and then two bands at the bottom, which is the white DNA, the white mouse. Now, if we can do genetic engineering in this oocyte, and we can remove the part of DNA that we don't like. Let's say we remove the bottom part. And then all the DNA, or not all, but most of the DNA of the mitochondria will now be of one type. You don't need to eliminate all the DNA. In fact, if we haven't eliminated all this DNA, you can see a faint bands just below to the left, but that's enough because as you can see here, if we take this oocyte, fertilize, now what happens is that this black and mouse give rise to just one color mice, which is white. There is only one mitochondrial DNA that shows the phenotype, in this case, white. So this is a clear example that we can modify mitochondrial DNA at will, and therefore we could modify any mitochondrial disease that has really a dramatic phenotype in the child that inherit this mitochondria. We can not just modify mitochondrial DNA, but we can also modify nuclear DNA. And last year we made the proof of concept in this mutation is these people that they die of a sudden death. They seem to be healthy. One day they are running and they just drop in the floor and they die. And that's because they have a mutation that causes this arrhythmic effect in their heart. So what we did was to take embryos from these individuals, and using gene editing technologies, we were able to demonstrate in vitro that now we can correct that particular mutation and have an embryo where all the cells, in a big percentage of the cells in this case, are now wild type and don't have the mutation. These are experiments that were conducted mainly by our collaborator, Shukrat Mitalipov. And if you look at these embryos, you see that they look perfectly normal. What we didn't do was to implant these embryos back in the mother. We just analyzed that the correction was made. We let the embryos develop in the Petri dish, and that was it. I'm sure you are aware a couple of months ago that the first 2 babies where gene editing have been done in the embryo have been born, and this certainly raised the question of whether, not that this can be done, because this has been done, whether we should do it, both the mitochondrial and nuclear gene editing. I'll come to this topic at the end of my talk. This is just to show the feasibility and where we are at this moment that we can modify our genome at the earliest stages of embryogenesis. And therefore, one could think of trying to address mutations that correlate with some of the diseases that you see here, some hereditary cancers, Huntington disease, etc., and etc. And I'll come back to the appropriateness of this at the end of my talk. Now, but by and large, the problem is not at the embryonic stage. The problem is when we are adult, and that's when the disease manifests and when the aging takes place. In the last 30 years, molecular biologists have developed these technologies for gene editing, with the only caveat that these only work in cells that divide. But cells that do not divide, we don't have technologies to modify the genome. We need the cell to divide so that we can then modify the genome. But if we were to take a picture, what we say BRDU stands for cells that divide, of this rune here, we will see that it's quite red, the rune. This BRDU is red, because many of the cells at the adult stage do not divide. Many of our cells do not divide. So we need to develop technologies to modify the genome of cells that do not divide. And I'm going to show you one example that we have developed in the lab, and for that we use one of the tissues that is most commonly known as non-dividing, the neurons and the brain. Here we have introduced, I'm not going to go through the details of the technology, but we can modify, and you can see this, the genome of a neuron by knocking the GFP, the green protein. This shows that we can do gene editing knocking in cells that do not divide in an in vivo brain. How can this be applied? Well, mutations like this, Retinitis Pigmentosa. This is a mutation in the retina that causes little by little the blindness of the person that has it, and that correlates with aging. We can take the eye of an experimental model that produced, the wild type produced the protein in red. You can see it here. The mutant eye will not produce the protein and will make the organism blind. And now we can edit cells that do not divide in the retina, and you can see in red on the left side of the screen how the protein is now coming, and that will allow the animal to see again. And this can be used to practically, and can be applied to practically any of the more than 10,000 mutations that correlate with diseases in humans. For instance, we are now doing this with spinal muscular atrophy, which is the most excuse me, is the most common genetic cause of infant death. And you can see here how in the spinal cord now, and you will see this by dots, these green dots, we can gene edit in the mutation so that the muscle of these affected people can move again. And as I said, this now can be extended to any tissue that the most predominant component is non-dividing cell, the heart, the muscle, etc. But I would like to retake. Can I Take a little bit of water. Thank you. Excuse me. But I'd like to retake the topic to what the title is. How this helps us understanding aging, besides fixing specific mutations. Well, let's go for this mutation. These kids have a mutation in the protein that is the major component of the nuclear envelope, lamin A. And as a consequence, this lamin A mutation gives rise to a nuclear envelope that you can see on the left of the screen that is distorted. And this is the cause of this phenotype. These kids, which the oldest here is about 12, 13 years old, they look old. And they're going to die in 2 or 3 years. And they like of an aging disease, cardiovascular disease. So we can use this technology that I just mentioned to fix the cells of this mutation. And here you are going to see that done in an animal model. So this is a mouse where the mutation is the same. A mouse in the lab lives for 2 years. This is a mouse where the mutation has been created and it just lives for 3 or 4 months. And it has all the symptoms of an accelerated aging. When we use this technology, and it's just a small injection in the tail after the animal is born, here you see the 2-liter mates, and in a moment you are going to see which one was injected with this technology and which one not. We are now at 3 months. The animals are going to die, and the one that doesn't move will die tomorrow, but the one that was injected is now happy, living. Thank you very much. Thank you. So it's a huge difference. It's life or death. You can use these technologies to fix mutations that affect aging and have an animal that has now an increase in lifespan. But besides the genome, being able to modify the genome, you know that there is another layer of controlling the expression of our gene, and that's the epigenome. These methylation marks, these histones that decorate our genome, are key for the process of aging in particular. And I like to give this example. About 100 years ago and now, there is an increase overall of 25 years in the human lifespan. But if we were to look at the genome of a person 100 years ago and the genome of a person today, the genome is the same. What has changed, whatever we have done, affects the epigenome. If you look at an ant colony, you see the queen and the others. They have the same genome, but one lives much more than the others. And this is just the environment, this is the epigenome. So the epigenome has a big influence in the way we age in our disease. So trying to develop techniques that can modify the epigenome may help the process of understanding aging. And the most famous modification of the epigenome, perhaps, is this experiment that was done 14 years ago by Dr. Shinya Yamanaka, where he took an adult cell, an old cell, applied these 4 factors, these 4 genes, and converted this old cell in a new cell, in a young cell, in an embryonic stem cell-like. And what these 4 factors do, they remove all these epigenetic marks from the adult cell. They don't touch the genome, they affect the epigenome. And that modification makes a cell be young again and be able to express the full potential of an embryonic cell. We did this experiment in an in vivo animal, and again we used this accelerating animal that has the mutation, lamin A mutation, that I remind you these animals will not last too long and will present all the symptoms of an aging animal. So here we are not fixing the genome like before. The mutation is still there. All what we are doing is adding these 4 Yamanaka factors. So this mouse still has the mutation. We're just changing the epigenome. And changing the epigenome gives rise to a mouse that rejuvenates, that lives longer, that is a totally different mouse, where all the organs start to work much better again, and that increases lifespan. Even though that mouse still has the mutation in its genome that caused this accelerated aging, altering the epigenome of this mouse makes for this mouse live longer, younger, and we have better quality of life. So this can be extended to wild-type animals. I'm not going to go through that. Before I go into this, the implications of this is that no matter how we come from the factory, no matter what our parents gave us in our genome, changing the epigenome has a big influence in the aging process of our cells. And by altering the epigenome, we can alter the process of aging. There is another observation that I'd like to share with you, and that has to do with time. As Valerio was mentioning, time is perhaps one of the key events in the life of a cell. It's about, 4.5 billion years that life was generated on Earth. And every cell, every animal somehow depends on the sun. And throughout evolution, and there was the talk a couple of days ago by Dr. Paolo Sassone Corsi that probably explained much better this concept here, the importance of the circadian rhythms for our life, and I'm going to show you for our aging as well. So about 70% of our genes have this circadian rhythm. So they change their expression pattern during the day according to the light. Now we know that if we look at a young animal and an old animal, their circadian rhythms are very different. A young animal, a young person, it has these peaks. You can see these rims, these waves, up, down, peaks and valleys to the left, very clear. An old animal, these peaks are not that pronounced. You can see it here to the left. And this gives rise or is correlated with alteration in the metabolism of this animal. Now, what we have done is to alter these rims, and we have done this by using these reprogramming technologies. And using these reprogramming technologies, what it makes is that this old clock can now revert to a young clock. And probably this is behind the reason of this rejuvenation that we see in these animals. This is just a graphic to show this. And we are now trying to go into the details of the molecular interactions that take place during the reprogramming and the genes that control these circadian rhythms. So what I have told you is that by reprogramming a cell, by changing the epigenome, we can alter the process of aging. And sometimes I get asked, great, why don't I take these 4 factors and I become younger? And obviously you cannot take these 4 factors. They induce proliferation and they may induce tumors. We have to find ways to substitute for these factors in a safe manner. And I'm very happy, and I'm saying this for the first time, and this is a collaboration we are doing here between the Salk Institute and CAUS with the team of Pierre Magistretti, where what we're doing is modifying the metabolome of a cell so that now it can be reprogrammed. And here we can see how an astrocyte can be reverted back to a neural stem cell or to a neuron. And we can make a reprogramming of a cell into an earlier one so that the organs may rejuvenate with the use of just small metabolites that the body produces naturally and will certainly not be as harmful as modifying with the 4 factors. So I hope that on another occasion we will have time to detail all this experiment and how we could benefit from this knowledge. But this removal of the epigenetic marks with the 4 factors is something that is not very fine-tuned. You may want to remove a specific mark and not all the marks of the genome. There will be marks that are good to remove, other marks that are good to be added, etc. So you need to fine-tune this process, and that's what we are developing technologies for that. This is one example, and is using CRISPR-Cas again, but in this case, we eliminate the ability of this enzyme to cut the DNA. It just goes there into the DNA, and then we add a transcriptional activator, a transcriptional repressor, and that will alter the histone landscape of that part of the genome. And how could this be of practical use? Let me give you an example, a couple of examples. Let's say muscular dystrophy. Muscular dystrophy is caused by a mutation in the genome. We can either fix the genome with the techniques I have mentioned before. The problem there is that we need to cut the DNA, and when we cut the DNA will have to be closed again, and sometimes this introduces new mutations. Or we can use technologies like this, where we don't cut the DNA, we modify the epigenetic landscape, and as a consequence, what you see here is that this mouse on the left, that is the muscular dystrophy mouse, where the muscles are not working, now the muscle can work again, even though the mutation is still there. We have modified the epigenetic landscape in just that locus, not like with the reprogramming that we eliminate all the epigenetic marks. We have just modified a particular locus, and this mouse, even though it has that mutation, recover all muscle functionality, and the phenotype of muscular dystrophy is gone. Let me give you another example. Many times you have a disease that is not due to a mutation. Is that last night we went into a restaurant, we ate something a toxin that was bad for our kidney, and we developed an acute kidney problem that could end up in the death of the individual. So we could use this technology without modifying the DNA just to go after this problem. And here there is the example. If we use this toxin, cisplatin, and put it in the kidney, all the animals will die after 3 days because of an acute kidney failure. If at the same time we modify the epigenetic landscape of some genes, of some loci, for instance, inflammatory genes, or genes that are normally involved in making nephrons, what happened now is that these animals in green that after the cisplatin die after 3 or 4 days, all of them, after activating these genes, now 50% of them, they recover and they don't die. So this technology can not only be used to fix mutation, but to fix general deterioration process like could be a kidney malfunction, heart malfunction, etc. The advantages of this technology is that you don't cut the DNA. It's not a permanent thing that you do in the genome. It's something that will disappear at will, and that it can be multiplexed, that you can do it with many genes at a time. We're using this not to go after specific diseases. We're using this in the lab to try to reverse aging. And we are using different factors and alter the expression without cutting the DNA, just the epigenetic landscape of an old mouse to try to revert that to a young mouse. And Finally, and that's my last scientific slide, is that I mentioned to you a key component in our epigenome is these methylation marks that decorate the genome. And this is perhaps the most stable way to modify the epigenome. And about a year ago, we were able to show that we can specifically induce de novo DNA methylation in a cell. What is this good for? You will ask yourself. So there are many imprinting diseases early on in our life that could be fixed by altering the methylation landscape. The disease comes from an aberrant methylation, and technologies like this could be used to fix these early imprinting diseases. But this was proven in a cell in the lab in an in vitro situation. What we want to do this is in an in vivo situation, in an in vivo animal. And here is the first animal, to my knowledge, where we are proving this, where we can alter the methylation pattern of an animal, and that can be transmitted to the next generation. This will not only help to address some of the imprinting diseases, but it may help to try to better understand this old question in biology, that some epigenetic information may be heritable to the next generation in our life. And that's a key question for those of you that are interested in this topic that has not been resolved. And having technologies like this certainly will help. But we are using it, again, to try to modify and understand the methylation patterns of an aging organism and how we can modify that so that we can reverse aging. You know that there are methods that are supposed to, regimes that are supposed to slow down the process of aging, eating less. There are even a clinical trial going on with metformin that affects one of the pathways implicated in aging. There is several compounds that have been linked to aging, and we're trying to see what is the methylation status in specific cells of the body of these regimes, and see how we can manipulate that and demonstrate a direct effect of methylation and aging. So I'm finishing. What is it that I have told you today? I have told you today that we take this mouse, we put it through this revolving door, and either through cellular strategies like the chimeric approaches, modifying the epigenome, modifying the genome, reprogramming with these Yamanaka factors, we get a healthier and a younger mouse. That's great. That's wonderful, and keep doing that in the lab. But we are here not to cure, not to slow down the process of aging in a mouse. Obviously, we are here for another reason, and it's try to better understand this process in human. And I feel that in order to translate this from the lab to the clinic, still there's a long, long road and a long bridge that we need to cross. We talk about innovation. Let's do this and let's apply it tomorrow. I'm afraid that at least in this field, we are not there yet. We are still at the very basic aspect of the knowledge. And even though all of us, and I see a lot of enthusiasm in this country, in this university, to try to innovate and go immediately and obtain a product that can fix a problem in the clinic tomorrow, I think we should all realize that first is trying to understand the process better. And even though I have shown you slides that look really optimistic for trying to understand this process and make the proof of concept in the mouse before we can really go forward into the other side of the bridge. We need to solve many problems. And this idea of innovation cannot come overnight. We really need to dedicate time and support to basic science. Concrete aspect, efficiency and safety. Of course, I'm showing you the best picture I have in the mouse. But that doesn't mean that the efficiency is high. Efficiency still is very low. So we cannot go tomorrow to the clinic. Safety. There are many issues regarding safety, manipulating the genome, the epigenome, or the chimeric approach, or any other approach. So these are practical things that will take still knowledge and support before we can start crossing the bridge. Another aspect, we love to say, we are going to collaborate with this hospital and we're going to fix this problem. The reality is that the doctor is very busy trying to deal with the disease and the patient that he has or she has in front. They are at the clinic. And we are in the lab trying to gather this knowledge. But the dialogue is not existent. And even though we can force it and we can implement it, if we don't support it, it will never happen, and this crossing the bridge will take longer. And a last aspect is the legal and ethical components of the type of experiments I have introduced you today. I have mentioned about using monkeys as growing organs. And some of you, I could sense immediately that were a little bit, what? We're going to use a monkey to grow a human heart? This could not be done. It's not just the ethical aspect. Science is going always faster than regulation, administration. And we, as scientists, need somehow to educate our representatives, so to tell them what is it that we are doing so that they can really, not in an ethereal manner, we need to do innovation, but really, step by step, help and support crossing of this bridge. And my last slide is going back to one of the most important person in the field of biology that we have had, and this is Darwin, that clearly said that everything that happened since life started on this planet occurs by random mutation and the natural selection of this mutation. The summary of what I try to tell you today is that we don't depend on Darwin today. We, in the lab, we can really avoid this randomness, and we can read, write, or erase in the 2019 our genome and the epigenome. We don't depend on Darwin. And therefore, in reading through Darwin, I like to finish by saying, and this sentence he put there, it is not the strongest of the species that survives, not the most intelligent, but the one most responsive to change. And this is what we are here for. And I'm really pleased to establish this collaboration with different members at CAUS to be responsive to the changes that are taking place, but at the same time be realistic. This word of innovation, just that it happened tomorrow, it will be difficult if we don't go into the real problems I just mentioned. And the most important slide is the people who have done the work that I have mentioned. This is my team at the Salk Institute, and I'd also like to thank some of the members here at COS that we are collaborating with to develop some of these aspects. Pierre, Magistretti, Valerio, Mo, Antonio, etc. Thank you very much for your attention.
Thank you very much. Should work. Okay. Thank you very much, Carlos. Great. Highly inspiring talk. The session is open for questions. Over there. Anybody providing this lady with a microphone? No. We know you can speak up, but it's not enough.
Hello? Yeah.
Go ahead.
Thank you very much for your talk. I was wondering, you showed the results of the treated mice that had the disease, but what about healthy mice that got the treatment? I was wondering, do you have side effects or maybe improvement in their muscles or in their time of aging, like their longevity or something like that? Thank you.
Yes. So we haven't tested the lifespan of these mice, of wild-type mice, because it takes a long time. We're in the process of seeing those results. But when we fix our attention into particular tissues, and you mentioned the muscle of an old mouse, so if you injure the muscle of an old mouse, an old person, it doesn't regenerate. If at the same time that we injure the muscle of this geriatric mouse, we activate partially these 4 factors, you see that the muscle regenerates. So this also works for wild-type animals, but the lifespan increase, the experiment is in progress, and still I don't know the end result.
Next.
Well, thank you for your talk, and I guess I will have a comment and perhaps a few questions. So the brief comment is that in general, if the carrying capacity of the planet is X and the current life expectancy is 71 years worldwide, then if you double life expectancy, then the carrying capacity halves. And we seem to have quite a few people around the planet. So that's one comment. The second comment, if we are going that route of having multiple animals delivering organs to people, then obviously that technology, in my mind, would be geared to the very affluent, which then raises the reason to my question. So thank you for mentioning in the last bullet of the last second to last slide ethical issues, because I think that ethical issues are up front in most of the things that you talked about. And I was wondering what are the sources of funding for your nonprofit organization, and how do they deal with the ethical issues that would be upfront, with most organizations that belong to public or private academia. So that's one question. The second question, you mentioned Darwin, and I certainly am not a geneticist, but you didn't mention Mendel. And there is a big discussion now among prominent geneticists about the very definition of genes and about what CRISPR does or doesn't do and the damage that it does in interventions. And so the whole picture you painted, according to my prominent friends from the States, is not as clear as you make out it to be. So could you comment on that? So 2 issues. One, ethics, sources of funding, and the second one, just the issue of gene definition and then the CRISPR interventions. Thank you.
Thank you for bringing up especially the first comment and the first question. And this was the purpose, perhaps the most important message that I wanted to put in the table, and you have repeated it. We are in front of a revolution of methods that could change the evolution of our own species. And you mentioned what are we going to do if we live double, and how this is going to affect our own species. These are certainly major topics that we as a society need to address, and this is what I wanted to put up front, that the technology is coming, and we are not prepared to solve the type of questions that you just put very elegantly. How do we deal with this observation 2 months ago of 2 babies being born after the manipulation of their genome with no approval from any government, no agency? This is happening in the lab, and the technologies are there, and our governments need to establish the legal regulation by which we should or we shouldn't do things in this field, because I'm totally convinced that we are at a stage in human evolution where we can change our own species. And I don't know what to answer to you, because I myself introduced the experiment of modifying the genome of an embryo that has this sudden death mutation, and I said that we stop in the lab because we have a strict regulation of what we can do and we cannot do. But that doesn't mean that the neighbor that doesn't follow that regulation cannot do it, and this constitutes a major problem that we need to get together. I have to say that in the last 3 years, I was part of the National Academy of Science in the States, tried to deal with some advice and regulation to give to the government of how to deal with this. And my experience was that they are not listening. They have other more important things to do. And that's what worries me, because these problems are right now there. And I totally sympathize with you, and this is one of the major questions we need to ask ourselves. We scientists are here to uncover and leave the rock and see the problem, but the entire society needs to chip in to see how do we go forward in that thing that we have discovered. I don't know if that answered your comment, but I totally have the same.
That's actually a good answer. It's an incredibly complex and vexing problem, which in how humanity subverts every good idea will lead to probably some very bad things happening around the world, and perhaps especially in China, where there is no regulations and you can do whatever you want. So yes.
Mm-hmm. So regarding the funding sources, there are experiments that are funded. In our case, we work in the States, and normally is the NIH the major source of funding. That is okay in the in vitro stages. There are other experiments that the regulations don't allow this. But independently of where the funding comes, with many of the time it's just donors who give the money unrestricted, we have the protocols that we need to follow and obey. And that's our main dictation. So there are experiments that we will never do because the protocols are not there yet, are not approved. But that doesn't mean that another lab cannot do it, independently of the protocol. So the problem is there, and we need to deal with it properly. And then I wanted to put a glossy picture. You mentioned the problems that the use of CRISPR can bring to the genome. You cut the DNA, and the moment you cut the DNA will try to close again, and mutations will be introduced. And maybe these mutations are nothing, but maybe it's even create a problem bigger than the one you wanted to fix. And we don't have enough knowledge still to somehow to make sure, and that's why I mentioned safety, to bring this to the clinic. We need to know, because after all, what CRISPR and many other enzymes are doing is just cutting. It's not that they are doing anything, they just cut, and this allows us to modify, introduce things there. So I mentioned other technologies where you don't cut the DNA, and that's modifying the epigenome. And it's not a permanent action. It can be removed after you have fixed the problem. So I think technologies like this, where the DNA is not cut, where gene expression is altered in a temporal window, could be safer and could be more practical. But still, we are at the very beginning of this technology, both in the genome editing or epigenome editing, and that's why I insisted that even though there's a lot of pressure by companies, by some governments, innovation, application of this, what you do, let's bring it to the clinic. I think we should be prudent, we should be cautious, because still we don't have enough knowledge, and the basic science still needs to learn a lot.
I'll take it offline. We can discuss it later. Thank you so much.
If I may add a comment to this, I mean, it may sound trivial, but as you mentioned, we are at the very beginning of understanding fundamental processes. Now, the idea that then we have the keys to do it right now is probably the most dangerous one. And this means that we know what we need is more and more resources to go deeper into the basic knowledge, produce those new type of concepts that can drive the next step. And this is probably the challenge, taking more responsibility from the society, from the governments and whatever, in producing more fundamental knowledge rather than accelerating a process that would end up in the probably dangerous perspective in a way. Question over there?
Hi, so thank you for the nice talk. I wanted to ask about, so we have these animals we know are not susceptible to change, to aging, such as tardigrades. And my question is, what is the possibility or usability to take their DNA to learn from how they do not age and to apply that to the human DNA? Is that possible at all, or is that not really something?
I'm trying to see if I grab your question. So what are the changes? Can you repeat again so that I can properly...
So we have an animal such as a tardigrade. So that animal, it does not age. It's such an animal that it does not die due to age. It's not susceptible to aging.
Well, it is. It's just the process is slower.
Slow down.
Okay,
Slow down. We're going to die, and we're going to age. This It's just how we can slow down the process and what are the parameters in the genome or epigenome that modifying it can slow down the process.
Well, essentially, yeah. To mimic such as a tardigrade or any other animal that has managed to, seemingly slow down the aging process.
Yes, I didn't have time, for instance, one experiment we are doing. Let's see, in rodents, there are different species of rodents. There is the rat that in the normal life in a lab lives for 2 years. There is this naked mole rat that is very close to the rat that we know, lives for 30 years. So we are trying to compare and modify the genome and the epigenome of these two animals on the knowledge we are acquiring to try to understand why these two close species have a different aging process, so different, 2 to 30 years. So this perhaps goes to your question, what are the differences that slow down aging? But for that we need technologies. And this is what I try to mention today. Technologies that could help us to understand this process. I'm not sure if I have answered your question, but maybe later with time I'll try to answer it better.
Okay, thank you.
One more question here in the middle. Can launch the right.
Hi. First of all, thank for the beautiful lesson. So I have two questions. The first one is there a threshold in the percentage of non-host cells that you can use to produce a chimera? And the second question is, you say that producing a human organ in a pig is not feasible because there is a kind of gap, crosstalk between pig cells and human cells. So I was wondering, would this efficiency increase if we try to produce the human organ in a pig-human chimera or pig-monkey chimera? And I have a third question, if possible.
Let me answer these two because I forget the third one.
Yeah.
This costs more, the third question.
So
I will pay.
Okay.
the second question is a very good experiment. We have not done it. So that's a very good idea, and we haven't done it yet, so I don't know. The first one, the number of cells. If we put over 10 cells, development doesn't proceed, so it stops. If we just put 2 or 3 cells, the percentage of chimerism is too low. So we're still trying to see what is the right number of cells and why the process is different when we put a certain number of cells. And the third question?
The last question is the missing factor, let's say, allowing the crosstalk between cells from two different species. Do you think it's a circulating factor that we can find in the bloodstream, for example?
No, it's just alterations in the timing of signaling. For instance, you have a receptor and a ligand. They are conserved in the mouse, in the human, in the monkey. But maybe at the time they interact during embryogenesis is slightly different. And when we mix the cells, they are not in sync and they cannot see each other. So just by altering, for instance, the timing of expression of, say, the TGF-beta pathway of the ligand and the receptor, we can see now that the dialogue is better.
Okay, thanks.
You mentioned the fact is, the impression we have, everybody has, is that these are very complicated processes. Using even different animals that can host or reproduce, whatever. But can you comment also on something you discussed earlier this morning on a more reductionist type of approach? So can we create a synthetic environment where we can use to accelerate the knowledge and maybe even go for a more applied perspectives?
Mm-hmm. Thank you, Valerio. This morning we were discussing, I didn't have time now, on alternative approaches to understand better these chimerism problems that we have. And I raised the example of synthetic embryology. This is a new area in biology in general. Synthetic, something that is exclusively done in the lab, coming from the knowledge we are acquiring and, say, applying, if we want to call it artificial intelligence, if we want to do this, and the predetermination that the cell has. And the idea comes from the fact that we just take one cell, an adult cell, we put it in the Petri dish, and we can generate, at this moment, what we call a blastoid and an embryoid, which meaning an embryo, which is not a natural produced embryo, it's just an adult cell. But by changing certain parameters in the media culture, really we can apply all the knowledge we are acquiring to produce, it's not an embryo, but it's looking like an embryo that implants and look like a synthetic embryo. So this raises the question that Valerio wanted to address. Where is this going? We now are in front of technologies that the knowledge can help us to produce a living organism, maybe in the future, just starting with one hair, and how this is going to inform technologies like the chimera to create organs. I will need a little bit of time to explain this, but I thank you for raising this comment, because this is another area of biology, not just in the production of organs, that I certainly think will have a big impact in our life in the near future. But I want to go back to the comment of Valerio, the importance of supporting basic science. We are still very early on in the game and we know very little.
Thank you very much, Juan Carlos. Any more questions from the audience? If not, Juan Carlos, there is a surprise here. That's always hidden here. It's actually a gift which we give to our keynote speakers. May you open it? It's an important contribution.
Labio Orientale.
Particularly fitting also the theme of the WEP with the Astrolab.
Thank you very much, Valerio.
I would like to mention the fact that, maybe by no chance, I think now, the Times Magazine has nominated Juan Carlos Belmonte as one of the 50 most influential personalities in now medical health perspectives. But as you can see, the importance of whatever nomination an influential magazine may have, the importance is to have the wisdom of a scientist bringing the right word and bringing people to reality and what is possible, what is not possible, or what our responsibility is. Thank you very much.